All right, students, we are moving into the next lesson in our central dogma unit. In the previous lesson, we talked about how DNA is copied in the process of replication. In this lesson, we're going to talk about how DNA can be transcribed into RNA in the process of transcription. To review here, remember our central dogma of life is DNA to RNA to proteins. Now, we talked before about how DNA can be copied in the process of replication. We also see two other steps here, transcription and translation. In transcription, we're going to be taking that DNA and turning it into RNA. And in translation, we'll take that RNA and turn it into a protein. And our ultimate goal here is to make proteins. Proteins run the show in the cell. We need proteins to carry out many functions important for life, and we're able to make those proteins by using DNA as a blueprint. However, remember that DNA is found in the nucleus of the cell, and it's actually going to be too big to leave the nucleus, and our proteins are made in the cytoplasm. So in order to get DNA out of the nucleus, we have to make a smaller molecule, which is going to be RNA. DNA is going to be converted to RNA in the nucleus, and this will be our process of transcription. Then that RNA can be read in the cytoplasm to make a protein, and this is going to be translation. And translation will happen in the cytoplasm. So we can still think of RNA like a blueprint because it's carrying a sequence of bases which will have instructions needed to make a specific protein, which is our ultimate goal, is making those proteins. And we're going to focus on transcription first in this lesson. Transcription is the process of converting DNA into RNA. Our key enzyme here is going to be RNA polymerase. So this process is going to look pretty similar to DNA replication, but this time we're only going to have one enzyme instead of those three enzymes we saw in DNA replication. So RNA polymerase is running the show here. This process will occur in the nucleus of the cell, and just like we saw with replication, this will occur from 5' to 3'. So essentially, we're using this DNA as a template in order to make a molecule of mRNA. Now, DNA is being converted into RNA in transcription, and we're actually going to have a few different types of RNA that can be made. So remember, RNA stands for ribonucleic acid. And this is a single-stranded molecule. So we can see our three different types of RNA here. Our first is mRNA. Now this M is going to stand for messenger. So our mRNA molecule, which we can see down here, is going to be our blueprint for translation. This will contain the instructions needed to make specific proteins. Another type of RNA that we can make is rRNA. So this R stands for ribosomal RNA. And this type will combine with proteins to make ribosomes, which we can see right here. So this ribosome here is rRNA and proteins mixed together. Lastly, we can make what we call transfer RNA. So this is our T here, transfer RNA. And this is going to bring amino acids to the ribosome during translation. So these are all going to be really important when we talk about translation in the next lesson. And in transcription, we're able to take DNA and form RNA. Now we're going to look at our steps of transcription. We're going to start with step 1, initiation. So our RNA polymerase is represented by this gray box here. So this is RNA polymerase. Our RNA polymerase is going to bind to the DNA at what we call a transcription start site. And this will start to unwind the DNA. So we can see we have this nice bubble here, and we're forming that template of DNA. Now once we have this template, we're going to be able to add some RNA base pairs. And this is going to happen during elongation. So RNA polymerase will add our complementary RNA base pairs to make a strand of mRNA. So this blue strand here is going to be our template strand of DNA. Let's say that these bases were A, G, T, C. And let's add our complementary RNA base pairs. So remember that RNA uses the base uracil instead of thymine. So when we have an A here, we're going to add a U instead of a T, because this is RNA being formed in red here. Our other base pairs would be C, A, and G. So in elongation, our RNA polymerase will continue to add those RNA base pairs, helping us form that strand of mRNA. And lastly, we have what we call termination. So our RNA polymerase will reach a terminator sequence on the DNA. Our mRNA will be released, and our RNA polymerase will be released as well. So this RNAP here, that's just an abbreviation for RNA polymerase. Now when this mRNA is released, it's actually going to leave the nucleus and it's going to enter the cytoplasm, where it can be used in translation to make a protein. All right, let's do some practice with our base pairing. Here we see a template strand of DNA. What I want you to do is add the complementary RNA base pairs. So go ahead and pause the video and give this a try. All right, this is what we should get. Remember that in RNA, we use the base uracil instead of thymine. So anytime we see an A in DNA, we're going to end up with a U in RNA. Let's do one more practice problem here. So go ahead and pause the video again and give this another go. All right, and this is what we should end up with for the second strand. Again, remember that RNA uses uracil instead of thymine. So to summarize here, we took DNA and we made RNA, this mRNA strand, in the process of transcription. Now in the next video, we're going to talk about how we can take this mRNA template strand that we made and we can translate that into a protein. So our next lesson is going to focus on translation.